Hi, and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at the factors that are going to affect an enzyme's activity. So we know that enzymes are globular proteins. We know that they act as biological catalysts and they will form these temporary bonds with substrates which are going to put a strain on the bonds within the substrate. So when we have an enzyme substrate complex forming, what's going to happen is that successful collision and reduction in the activation energy is going to result in the rate of the reaction happening at a faster rate. And that's why we call them biological catalysts. So what we have to understand then is when we look at the rate of reaction, what we have is if we increase the substrate concentration, we're going to result in having a higher collision rate. A higher collision rate until we reach what's known as the maximum rate of reaction, or Vmax. This is where on a graph you will see it start to plateau. Now it's important to note what we've got with this higher collision rate is more enzyme substrate complexes forming, and before what we had was that some of those active sites were actually free when there was a lower concentration of the substrate. So therefore the substrate concentration was limiting the rate of the reaction. Now as we get up to that Vmax, all those active sites have now become filled, and therefore it doesn't matter how much more substrate we give to the solution, what's going to happen is it will only work at that Vmax rate. So it's important to note that there are certain factors that are going to affect the rate of the reaction. Now, if we increase the temperature, we know that we're going to increase the temperature and therefore increase the kinetic energy of both the substrate and the enzymes. More kinetic energy, more frequent successful collisions, more enzyme substrate complexes are going to be formed. Now, normally we say that the rate will double every 10 degrees C. But you need to know what's known as the temperature coefficient, the Q10. And that is the increase in the rate of reaction with a 10 degrees C increase. So if I'm saying that the rate is going to double every 10 degrees C, you will be expected to prove that or to calculate that. So if they give you a graph, for example, they may show that the rate at 20 degrees C is 0.009 seconds. And if the rate at 30 degrees C is 0.017 seconds, then what you should understand is that your Q10 would therefore be your higher temperature, your R2, 30 degrees C, divided by your R1 temperature, 20 degrees C, and therefore that will give you an answer which is 1.88 reoccurring. So if we were to round that, that would then take us to 2. So in the exam, if they give you a graph, it is important that you use the numbers that are there in front of you so that you can work out your temperature coefficient. Now, the key thing to note is as we increase the temperature, we will get to a point where we will see a peak in our graph. That is known as the optimum temperature. So the optimum temperature is where the rate of reaction is at its highest, and therefore the highest number of enzyme substrate complexes are being formed and the rate of the reaction is at its greatest. If we increase temperature higher than the optimum, what we can see is that the rate of reaction actually drops down. And that's because what's happening is denaturation. We are increasing the temperature. That temperature is going to increase vibrations within the enzyme. Now, this is going to then cause bonds to break, which will affect the tertiary structure of the globular protein. This is irreversible. So as you increase temperature, after you have passed the optimum temperature, you will start to have that denaturation occurring. The more we increase the temperature, the more likely that denaturation will get to the point where it will completely stop the enzyme from being complementary to the substrate, and therefore that tertiary structure has changed. The active site shape is no longer going to complement, the enzyme isn't going to function, and the rate of reaction will drop down. So it's important you note how temperature has an effect on the rate of reaction. And what you also have to know is how pH will have an effect. Now, pH is determined by the availability of hydrogen ions. So if we are down with a low pH, for example, pepsin is an enzyme that works in acidic conditions. Now, if we have a low pH, we have lots of hydrogen ions present. If we're at a neutral pH, like trypsin, that is where we don't have lots of hydrogen ions which are free in the solution. So different enzymes have got different optimum pHs. And what we're talking about here is those hydrogen ions and how they're going to interact with the polar and charged R groups and therefore affect the tertiary structure of the enzyme as it moves away from its optimum pH. So wherever you see the peak, that is what we would class as the optimum pH. If we move away from the optimum, we lower or increase the pH. That is therefore going to affect that tertiary structure, affecting the active site shape, affecting the ability of that enzyme substrate complex to be formed. So this is a reversible denaturation. So what we tend to do if we're doing enzyme investigations is we tend to put in a buffer solution to ensure that we are not affecting the rate of the reaction by changes in the pH that may occur due to the breaking down of certain substances. Now, a protease is a very specific type of enzyme that is going to digest proteins into smaller peptides. So if we have trypsin, as we were just looking at, trypsin is a protease which is going to break our proteins down into smaller peptides. Then we have endopeptidase. This is another enzyme, another protease, and this is going to break our smaller peptides down into our amino acids. So it's important to note when they tell you in an exam, if it's a protease, then you know that that enzyme is going to break down proteins. So trypsin works at a pH of 7, neutral pH. That's its optimum. Whereas if we were looking at our pepsin, that we were just talking about before, that pepsin actually has an active form where it works at a lower pH, an acidic pH. So what you tend to have is an enzyme which is produced in what's known as an inactive form. So pepsinogen is an inactive form, a precursor enzyme. When the precursor enzyme is released into the stomach acid, that is then going to change its tertiary structure into the active form, which is known as pepsin. So the acidic pH of the stomach actually changes the tertiary structure, making that active form, and that way what the body does is it prevents any tissue damage from occurring because the precursor enzyme is only activated once it's in the optimum conditions, it's in the stomach acid. So the other thing you need to know when it comes to enzymes and factors that affect the rate is they will most likely give you a question that looks at an enzyme called catalase. So hydrogen peroxide is a toxic metabolic waste product, and we have got enzymes that are produced in both animals and plants that will break that hydrogen peroxide down into water and oxygen, non-toxic substances. So catalase is the enzyme, and that enzyme is very specific in breaking down that substrate, hydrogen peroxide, into the products, water and oxygen. So if we're going to do a practical on this, we would be looking at the rate of the reaction by calculating the volume of the gas oxygen that's being collected. So when they are looking at this, they'll be checking your practical skill application. So an experiment is going to be investigating the effects of a substrate concentration on the rate of an enzyme controlled reaction. So in this case, you need to be thinking about, well, what's my independent variable? So my independent variable, for example, would be the concentration of hydrogen peroxide. Now, always try to have key values to give, and I would say a rule of thumb of 5. 5 values that you would want to use. So in this case, my concentration of hydrogen peroxide is 2, 4, 8, 16 and 20 arbitrary units. Now, are they a good way of indicating the concentration of hydrogen peroxide and its effect on the enzyme controlled reaction? Well, this would be through using a zero dilution to get to those concentrations, but actually I might want to look at certain values in between. So that's another way where they can assess your practical skills in this type of question. What's my dependent variable? Well, my dependent variable is my rate of reaction. So the volume of gas given off every 30 seconds for 3 minutes. Do not forget that rate of reaction is the volume of gas that you are collecting over a specific time period. And then always think about those control variables. As I said before, pH is going to have an effect on the rate of a reaction, so you need a buffer solution. Temperature will have an effect on the rate of reaction, so you need a water bath to control the temperature. The enzyme concentration is going to have an effect on the rate of reaction, so therefore you will want to control the surface area and the mass of whatever your source of your catalase is. So for example, if you're looking at liver, you want to control the mass of the liver, but you won't be able to control the surface area of the liver. Whereas if you're looking at a potato, for example, you can control the surface area, and therefore that might be a better source of the catalase for this rate of reaction. And obviously remember your volumes. If you need to control the variable, if you are changing the concentration, you need to control the volume of hydrogen peroxide for each of your tests. So it's important that you're also making sure that the results you're collecting are valid, and to make them valid, to increase the validity in your data, you always want to repeat, and you need to do that again, rule of thumb, at least 5 times. Why? So that you can calculate a mean and standard deviation, and then you can graph out the data and conduct a statistical test. So the other thing you need to note then is about the introduction of inhibitors. So if we have a competitive inhibitor is going to have a similar shape to the substrate, and therefore because it has a similar shape, it's going to be a similar shape to the active site. So it's going to complement the active site of the enzyme. So therefore what you're going to have is that the competitive inhibitor will block the active site temporarily. So because the substrate is not able to bind, it's being blocked, the rate of the reaction will be lower. A non-competitive inhibitor is one which is going to bind to an allosteric site, and because it's binding to an allosteric site, it's going to change the shape of the tertiary structure. That therefore is going to result in the active site no longer being complementary. So if we look at the rate of reaction that occurs when we have our competitive inhibitor and our non-competitive inhibitor, what we can see is when there is no inhibitor, our rate of reaction will increase and then plateau. It reaches our Vmax when we are increasing that substrate concentration. If we have a fixed volume of enzymes and we have a fixed volume of our competitive inhibitor and our non-competitive inhibitor, what we will see is that our competitive inhibitor will decrease the rate of the reaction, but it will not change the Vmax. as their effect can be overcome by us increasing our substrate concentration. So what you should note with a graph is if the rate of reaction is increasing and getting close to, but hasn't yet reached the V max, that therefore means we might not have put in enough of our substrate concentration. With regards to our non-competitive inhibitor, is going to decrease the rate of the reaction, and the V max is never going to be reached. And that's because as that active site changes shape, it's making them unavailable. So they are never going to reach the same level as when there is no inhibitor present. So you can quite clearly see in a graph, whether you are looking at a competitive inhibitor, the rate has been decreased but is working its way up to that V max point, or if you're looking at a non-competitive inhibitor, the plateau will be reached a lot quicker. And the last thing to note is what's known as your end product inhibition. With an end product inhibition, this is something that's going to regulate the rate of a metabolic reaction by preventing the overproduction of the final product. So here we can see we've got oxaloacetate. We have an enzyme that's going to produce intermediate 1, enzyme intermediate 2, an enzyme intermediate 3, an enzyme intermediate 4, and another enzyme that reaches us to our end product, which is lysine. Now, our end product inhibition is a product of the reaction is going to act as an inhibitor to the enzyme that would produce it. Now, this ensures that resources that are not wasted. So therefore, you can quite clearly see that lysine is an end product that is going to inhibit my yellow enzyme, the enzyme that will turn intermediate 3 into product number 4, or intermediate 4. The lysine, the end product, also inhibits our blue enzyme, our enzyme that is going to convert oxaloacetate into intermediate 1. Therefore, this is an example of a non-competitive reversible inhibition. It's a non-competitive reversible inhibition because it's not competing for the active site. It is going to bind to an allosteric site. It is going to affect the active site shape, but it is reversible because it can be removed when the concentration of lysine has decreased. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. And if you haven't already done so, please do check out my revision platform, www.aiqchat.com, and that should help you to revise.